Hi, my name is Robin Hall and I work as the Project Manager and Outreach Coordinator for the Reactome project. Now, as part of the IDG program, the Reactome group has built a new tool called the Reactome-IDG Portal that utilizes the Reactome knowledge base to systematically illuminate interactions of the understudied or dark proteins with other proteins and biological entities in the context of Reactome pathways. In the next few minutes, I'll demonstrate some of the key features of the Reactome-IDG Portal. Welcome to the Reactome-IDG Portal. The logo at the top here provides a way to navigate back to the home page as you're working your way through the website. The link here to the user guide and a link to the download directory. You can access the Reactome-IDG Pathway browser directly by clicking this button, or continuing back to the main Reactome website by clicking this button here. There's a quick blurb about the project, and there's a search bar which allows you to query the Reactome-IDG portal. So as an example, CDKL5 is a dark protein. As you can see from the results, there are no annotated pathways found in the Reactome knowledge base for CDKL5. We can, however, link CDKL5 through functional interactions to pathways in the Reactome knowledge base. And so what we've done is we've mined a large corpus of protein-protein interaction data from TCRD, from TCGA, and from other interaction data sets to create this large functional interaction network. We can identify within that network CDKL interacting partners. We can create a gene list from that, and we can perform pathway enrichment analysis against the Reactome knowledge base to identify CDKL5 interacting pathways. You can see this list here. We can click on this link here, and this takes us to the Reactome pathway browser. The hierarchy here on the left provides a list of all the reactions and pathways known to Reactome. There's a diagram here on the right, and then there's a details panel below which changes as you navigate through the diagram or interact with the hierarchy. So as I zoom in a little bit more to look at the details of this pathway diagram, you'll see that some of the pathway nodes have this pink halo around them. And this corresponds to CDKL5 interacting partners. So it could be individual protein nodes, or in some cases macromolecular complexes where there's a CDKL5 interacting protein. The other color overlay that you can see within the node is representing the IDG target classification, the key of which is represented here on the right. Now we can toggle very quickly from the pathway diagram view into the network view by clicking this button. And again, we're seeing that same pink halo around CDKL5 interactors and that IDG target classification color scheme as well. We're just going to navigate back to the diagram, just zoom out a little bit here, and we can press this X to remove that pink halo. Now, one of the other additional features of this tool is the ability to overlay tissue expression data onto these pathway and network diagrams. And so we've mined 19 different data sets from the TCRD resource. We can access it through this pop-up window, just picking a data set and then selecting up to 12 tissues of interest. I'm just going to pick 5. You click the overlay button, and you can see that the diagram has recolorized according to the expression data set. You can navigate individually through the tissues using the navigator feature at the bottom here, or you can press the play button and you can put it on a continuous loop. You can navigate very quickly over to the network view, and again, you can see the color scheme changes. I can pause it, I can remove that, and then I can go back to the pathway view, and I will demonstrate the final feature of the overlay, and that is overlay pairwise relationship data. So we've again mined all of that interaction data, and we can overlay that onto the diagram or the functional interaction view. We start by selecting the type of data we'd like to show. In this case, it's going to be protein-protein interaction data, the source data, which is from BioGrid, Bioplex, and String. If there's positive and negative interaction information, we can colorize those edges specifically with these different color selections. We can add the data set, and we can add up to 6 pairwise relationships and overlay that onto the diagram. So we press the overlay button. Now as I zoom into the diagram a little bit here, you can see that on summon nodes at the top right corner, there is this appearance of a red circle. This illustrates the number of interactions. If you gloss over your mouse, you click on that red circle and you can see this pop-up window appearing with a network viewer. And you can see in the center is the RAC1 protein, which is in the diagram, and the triangle represents the functional interactions. Now there's too many functional interactions to show in a single view, so we provide this table with additional information about the pairwise relationship between the target protein, the pairwise interactor, the IDG target classification, whether this interaction is negative or positive, the source of the information, and also the relationship information, which can be viewed in a separate tab. You can also navigate through the data that's been presented in the table, or you can filter a query by gene name or data source or the type of the edge. If I move over to the network view, by selecting a protein like PTK2, you can click on it, right click, and you see this little halo here. It allows you to show pairwise relationship information, very similarly to as I showed you a moment ago, the network viewer here and the table of information below. Now one final little feature that is nice to show with this is the ability to view drug target information. So if we click the RX button here, we can now see the drug that interacts with PTK2, and we can get in the table some additional information about the drug and the drug target. Likewise, if we go back to the pathway diagram, we can see that there is a little blue circle at the top left corner. In this case, it's showing 8 drug interactions. If we click on that, we get a network view of the components of that macromolecular complex, and we can see the interactions with those drugs. And again, that table of results information below that. Well, that concludes my demonstration of the Reactome-IDG portal. I'd like to take the opportunity to thank both the core and IDG Reactome teams, our grant funders, and our IDG collaborators.